right now. Okay, great. So many of you know Bob, but I'd like to introduce him for those of you who don't. Dr. Robert Michael Pyle is a charter member of the Washington Butterfly Association, and he is the author of 22 books, amongst them titles that are familiar to many of you: Watching Washington Butterflies, The Butterflies of Cascadia, and with co-author Caitlin Labar, The Butterflies of the Pacific Northwest. Bob is the founder of the Xerces Society 50 years ago now, and was a pioneer of butterfly conservation, watching, and photography. He is the coordinator of the Washington Butterfly Survey with John Pelham, and he is also an honorary fellow of both the Royal and American Entomological Societies. I want to make special mention of these last accolades because there are only 8 scientists who have been awarded both of these honors. And to give you an idea of how fortunate we are at Washington Butterfly Association, our next month's speaker is also a doubly honored fellow, and that's Dr. Lynn Riddiford, who will be giving a presentation for our December meeting on metamorphosis. She is an emerita professor at the University of Washington, located in Friday Harbor, and we're very much looking forward to having her with us as well. But this month we have the other doubly honored fellow who will speak to us, and that is Bob Pyle. The title of his presentation is The Mariposa Copper Butterfly in Washington and Beyond. And thank you so much for being with us tonight, Bob.
Thank you, Paulette, and thank you for that very kind and thorough introduction. I'm now going to share my screen. Does that work for everyone?
Yes.
You have my shared screen. Good.
Yes.
Well, thanks for having me back to speak to WABA, an organization that's dearly close to my heart, and a lot of you are too, a lot of good friends here with us tonight. Thank you for coming. And it is coming. It is an effort, even if you don't have to get into your car and drive somewhere. I know it is. We all know it is. So I hope that you'll receive some enlightenment, perhaps, but possibly entertainment as well, and some curiosity. And I hope that at the end of this talk, you'll come out with a little bit better awareness and knowledge and familiarity with one of our most beloved butterflies. It is, of course, the butterfly, because its name, Mariposa, means butterfly in Spanish. So it's the butterfly. And if you see one in Mariposa, California, and it does occur there or quite near there in west of Yosemite, you'll be triply pleasured because it's Thursalia mariposa at Mariposa, which is a Mariposa. Now I'll stop all this. Okay, let's carry on. I want to make it clear that this is a version of a paper that I co-authored with Paul Hammond. So I place him here as a co-author with me, and in a way a co-presenter, though he has not seen or approved this version of it. It's a big project that was a 40-year culmination of studies, and I could never have finished it without him and a number of other people, John and others, whom I'll mention at the end of my acknowledgements. Mariposa Coppers in Washington and Beyond, a review, taxonomy, and biogeography of Thursalia mariposa, named by Riekert in 1866. And an alternative common name for the butterfly in some books is Riekert's copper. You may know it under the name, and most of the field guides have it under the name Lycaena mariposa. But in the current Pelham catalog, the genus has been, an older name, Thursalia, has been used instead of Lycaena, which is an old world name. Well, okay, slide advance, maybe down here. Good. And is my cursor working for a pointer? Yes?
Yes.
Good. Thank you. The Mariposa coppers, one of our copper butterflies, obviously, most of which are not bright copper, although a couple of them, the ruddy copper and the lustrous copper, are. Most of our coppers are more the color of an old penny. And they might or might not have purplish reflections in the males. This being a male, this classic male copper. Many coppers have these orange zigzags or lunules in the submarginal area of the hindwing. And one of those is the Mariposa. So on top, you'd be a little bit hard pressed to say which species it is. Is this a Mariposa, or is it a purplish copper, or is it a lilac bordered copper? One clue is that the tips of the veins have dark scales, which makes the margin or the fringe of the margin checkered. And that's a trait that's more strongly developed in Mariposa. So if you see a top side of a dorsal surface of a male copper, look for the checkered fringes or not. Now the females, as with most coppers, are of a different form from the males. So the species are sexually dimorphic. And the females, as with the purplish copper, have a lot more orange. And some people mistake them for crescent spots or small fritillaries or even skippers. But notice the lycaenid or gossamer wing shaped wings and the orange zigzags again, which appear on the dorsal surface as well as the underside on these butterflies. And then again, the checkered fringes. So there's a classic male and female of the Mariposa copper. It's the underside that's so very expressive and individual and memorable for this species. I've been intrigued by it ever since I was a little boy in Colorado, because Colorado butterflies indicated just one record for Colorado, and that was dubious. And then a friend of mine, a young kid, to whom I sold my extra specimens for his allowance and euchred him out of his best specimens for trades and so on, useful kid, came back from Yellowstone with a Mariposa copper that he claimed he caught up by Fort Collins. And so for years, I reported that in the literature, and for years, I thought I had a voucher specimen that confirmed that the Mariposa copper lived in Colorado. But as years went by, I got to worrying because Eddie had been in Yellowstone on that trip too, and he wasn't the best keeper of data. Remember my column from last month in WABA in Gnome about the importance of records and being very clear with them. Eddie wasn't that clear with his records. And ultimately, I decided that the so-called Fort Collins Mariposa copper was probably mislabeled. And to my great shame and disgust today, I destroyed the specimen. And later, Charles Remington at Yale told me, founder of the Lepidoptera Society, said, "Oh, Bob, never destroy a specimen, particularly if it's doubtful." And so I wish I had not, and I wish I had pictured it in our current paper, because clearly it was a member of the Wyoming subspecies. Well, all this is to say that from that early day, that picture in Colorado butterflies, I was so taken and intrigued by this particular pattern, which doesn't repeat on any other copper or on any other butterfly that I know. I call it kind of a Morse code pattern because it's dots and dashes, dots and dashes. And that's what it reminds me of. It has zigzags on the underside, but they're not orange, at least not in the male and seldom in the female. So this is the butterfly we're dealing with tonight. Now, this butterfly has undergone a lot of changes in recent years in our understanding of it. And I'm glad to say, happy to say, proud to say, honored to say, humbled to say that I have something to do with those changes, along with Paul Hammond and a lot of other people who helped us. Here we're looking at a page, it's a hypothetical page from Jonathan P. Pelham's Meisterwerk, his magnum opus, his great catalog of the butterflies of the United States and Canada, a world-regarded document. I had an email the other day asking for illustrations from our paper from Belgium. And the first thing that our Belgian colleague did was to quote Pelham's catalog. It's known all over the world, in case you weren't aware of that. Known and used and admired and envied. Well, if this is actually a picture of the modern one with deletions, but the original of Pelham's catalog looked like this approximately for the Mariposa copper. Tharsalia mariposa, Tharsalia mariposa mariposa, the type subspecies, in other words, the first subspecies described from the Sierra Nevada by Riekert in 1866 when the species was so-called discovered. Subspecies B, Tharsalia mariposa penrosi, which was named by W.D. Field, Bill Field. He was lepidopterist at the United States National Museum at the Smithsonian for many years. And he wrote another one of my scriptures as a boy, which was The Butterflies of Kansas. I lived on the Kansas side of Colorado, so that book was very helpful to me too. But Field went to Yellowstone and came back with some specimens that were different from the Sierra Nevada ones, and he named this new subspecies penrosi after a woman who had collected them in 1938. And there was only one other subspecies known at that time, Lycaena or Tharsalia mariposa charlatensis, named by W.J. Holland, the author of yet another one of our childhood Holy Writs, which was called the Butterfly Book. I wonder if any of you still use it. John and I grew up using it a great deal. Holland named this subspecies from, as you might guess, the Queen Charlotte Islands, now known as Haida Gwaii. Bill Yake and John Shepard and some other among our number have been there. But charlatensis is, I think, is it still the only species or one of very few species of butterflies ever found on Haida Gwaii, and it's abundant there. John Shepard's done quite a bit of work on it. And so Holland named it charlatensis. Nowadays, if you look at the current Pelham catalog, which you may do anytime via the web, you will see that this species looks like this. Quite a bit of subspecies inflation, although not needlessly so, because all of these new names passed the Pelham gauntlet, which I can assure you is a severe and difficult and properly, challengingly scientific gauntlet to pass, which was the greatest satisfaction of this whole study, other than knowing nature better and getting some good names on things and honoring my late wife, Tia. The great satisfaction was that John Pelham and our other colleagues gave his imprimatur to all these new taxa. So as you can see, we've learned a lot, and this page more or less symbolizes the conclusions of this study and its accomplishments. The story goes back with me to 1975. At that time, I was married to an English woman by the name of Sally Hughes, who is back in England with a wonderful family for many years and has retired recently from teaching in Oxford for a good long while. Here in this picture, a recent picture, she is actually studying the eggs. She had located some eggs of the brown hairstreak butterfly. There's all kinds of volunteer lepidopterists in England that go out and look for hairstreak eggs in the wintertime, so they know where the colonies of these rare hairstreaks are. Well, it was Sally who actually instigated this entire study by getting up before me, which almost everyone I've ever been with in any capacity, camping, married, whatever, has done, because I'm not a big early riser, which is one reason, though I am a birder, I am more of a lepidopterist than I am a birder. Sally liked to get up early, and we were camped here at Alstrom's Prairie. I don't know how many of you have been up to these surprising prairies in Northwest Washington. We're near the tip of the state here, and in fact, near the tip of the coterminous part of the country, Cape of Lava being the westernmost point in the coterminous United States. Cape Flattery is up here. It's farther north, but not farther west. If one goes to Lake Ozette, at the edge of Olympic National Park, one may take a triangle trail called the Ozette Triangle down to Sandpoint and up the beach to Cape of Lava, the site of the very famous and important archaeological dig of a Macaw native people's village that was inundated by a landslide, became one of the most important digs in the state. And then back again to Lake Ozette. These are on boardwalk trails. But on the way to Alstrom's, you cross, on the way to Cape of Lava, you cross this Alstrom's Prairie, which was named for Pete Alstrom, no, sorry, Lars Alstrom, I think it was, the westernmost homestead in the 48 states. Burnt by him, part of that burnt by the Indians, as was Roos's Prairie, for the production of various root crops. And there's more to that coming up later on. I'll simply mention that fact now and why we were there. It was 1975, I was doing my doctoral dissertation research on the distribution of Washington butterflies and what they could tell us about the distribution of nature reserves as they already existed and where the gaps were in that system. That summer, I was traveling all over the state with my wife at the time, Sally Hughes, who's the founder, by the way, of the annual butterfly counts, the 4th of July butterfly counts, run by NABA now, and with my brother, Howard Footstone Pyle, and with my field assistant, David Shaw, who now lives in Issaquah. We were traveling all over in a VW Bug, believe it or not, trying to come up with more butterfly records all over the state to integrate into my database. It occurred to me that whereas the Olympic rainforest possesses few butterflies, wouldn't it be interesting to see if these open sunny habitats immured in the middle of the rainforest might have more or might have some? And that's why we're there. We walked out the boardwalk trail through the rainforest and came in good time to Alstrom's Prairie. It really does seem like a prairie. Now I'll fast forward to tell you that many years later, Sally and I had a contract with the National Park Service to revisit Alstrom's Prairie because another Lars Crabo, one of our colleagues who studies moths to great effect, he's up in Bellingham, had seen a fritillary butterfly there and reported it, and it was thought that it might be the endangered Oregon silver spot. And so we were hired on a contract for the Park Service to see if that were the case. We did studies there, transects, studies of the violets, and everything else, never found any fritillaries. Concluded that if there had been a fritillary there, it was probably a hydaspes. There was no evidence of the Oregon silver spot, but it gave us a chance to do a lot more work on the current study. Now I'll go back again to that day in August of 1975. We're camped here at Alstrom's Prairie over in the edge. Sally gets up before breakfast. David and my brother Bud and I were all sleeping. And she comes back. The sun's over the edge of the rainforest now. And she says, I found some butterflies on the prairie. Excuse me. So we go out there, and indeed, there are little brown butterflies flying over the sphagnum moss among the bog cranberries. And they were clearly, with their dot dash topography on their wings, Mariposa Coppers. But what was this? They were dramatically different from any Mariposa Copper I'd ever seen, with very, very light coloration of the outer half to third of the wing. And in many of the individuals, the light patches were islands between the veins, almost looking like the orbs of a fritillary. We thought this quite distinctive. And even on the fresh species, the forewing ventrum was very pale, not just warm. Even more dramatic than that was the fact that the females were almost white on top, or off-white, sort of a buff cheesy color. Very, very strange. And here you can see the effect I'm talking about of the islanding cell formation of the white out near the edge. Is so very distinctive for this butterfly. They were nectaring on the, at least trying to nectar on the blue gentians, and nectaring a great deal on Gentiana douglassiana, the little white gentian, as well as sorbus, the, what's the common name of sorbus? You can't unmute yourself and tell me, I don't suppose.
Mountain ash.
No, was it sorbus? That's not what I meant then. Here, I'll show you a picture instead. Yeah, sorbus is mountain ash, of course. Oh, well, I'll come to one later and you'll see what I'm talking about. Can't think of his name right now. Well, so we took back some voucher specimens under permit from the Park Service and compared them to the existing subspecies, of which, as you know, there are only 3. One of them is the type subspecies Mariposa mariposa, and it doesn't have those characteristics at all. It's kind of sandy or buffy below. The butterfly was not, in many respects, not at all like our butterfly. So we compared it to the Queen Charlotte coppers that were present in the collection at Corvallis. Very, very dark on top, but this butterfly is muddy and murky below. Has some white scaling toward the outer part, but not nearly as much. And it's a distinctly smaller butterfly and different in several other traits as well. But it was the closest, both geographically and physically. And then the third type, which was Penroseae, the Rocky Mountain subspecies, was being used at that time too for some of the upper Northwest butterflies, and then the type subspecies sort of being used for the Cascadian ones, though they didn't really look like it. Those were the only choices we had, and this didn't look like it either. I was a little bit lighter and browner and grayer than the Californian ones, but it still didn't have this distinctive white submarginal band and islanding, or some other traits as well. So I decided a long time ago that the copper that we found that day was probably going to be a good new subspecies. Now, mind you, it took me a long time to convince anybody else of that. For a long time, the state was interested in knowing that, and so was the Park Service, because they wanted to conserve it. They didn't know how to manage Alström's prairie. Let it all succeed back up to hemlocks and Labrador tea and other shrubs and trees in the absence of fire, or to maintain the fire ecology, which seemed in some ways to some managers unnatural, though it had been conducted by the first peoples there. But we knew there were bog plants there as well, and that it had true bog characteristics. And there was a strong argument for maintaining its bog and prairie characteristics, much more so if this became a new subspecies. So we were pretty sure it was. However, the best we could do for years was to get people to refer to it on checklists as Lycaena mariposa charlotensis, because that was already named. It's a big deal to name a new taxon. It takes a lot of work, and you better be darn sure you're right. And so we used that name for a long time. Also, as I say, physically, it was the nearest up the BC coast, and probably had some evolutionary connection to it, though it wasn't very much like it now. This went on for a long time until finally, going to the Northwest Lepidopterist meetings year after year, I took specimens to show them being my doubters, primarily John Pelham and John Shepard and others who wanted to get it right. I said, look at this butterfly. Look how different it is from all these others. And finally, I showed some of the very pale, they'd be even much paler, females and the very well-banded males to John Pelham one day, who'd had a lot of experience with charlotensis And he said, "Oh, yes, I see." And the penny dropped, and I got the blessing of John Shepherd. And John Pelham came to agree to that we did have a new subspecies. Consequently, Paul Hammond and I, years later, went to work on it. Now, I have to say, this paper never would have gotten finished if it had been just for me. I wasn't getting all the material mounted. I wasn't getting the comparisons done. Didn't have the material. And I also didn't have and do not have one of the characteristics that most exemplifies some of our finest naturalists. And that's what has always been known as the good eye. Oh, I get a pretty decent eye out there as an observer. But there are people like L. Paul Gray, who was Paul Hammond's predecessor as the king of the fritillaries. And Paul Hammond himself, John Pelham, Andy Warren, who have, and Vladimir Nabokov had it too, what's known as the good eye, the really good eye, to where you can see and remember the minute characteristic differences between different butterflies and remember them. It's kind of like Dennis Paulson's ear for birds, though he's got the good eye for birds too. Dave Nunnally has this good eye for the blues that he's worked on, but I don't have that. I'm not able to recall and remember and in my mind make these comparisons. I can do it when it's on paper or the specimens in front of me, but not the way they can, and Paul could. So Paul and I began gathering material from museums all around the country in the West, images and specimens. I went, for example, to the National Museum of Canada, and with the help of Jean-Paul Francois, the curator and his colleagues, brought back some hundreds of coppers from across Canada and the northern states. We had many in the Oregon Arthropod collection at Corvallis. There were many in the Burke Museum that John Pelham curates, California Academy of Sciences, Denver Museum of Science, many others. In the end, to make it short, we compared over 6,000 Mariposa coppers. If this offends any of you who dislike the taking of one butterfly, let alone 6,000, remember, these are covering decades and decades and decades, and many, many thousands of square miles. And in any one case, only a tiny number of specimens were taken from any population. And I can assure you that the impact that this collection as a whole would have had on the Mariposa copper is probably about tantamount to the impact that you had on the mosquitoes slapping them the last time you went to Alstrom's Prairie in the summertime. Collections, on the other hand, have a very powerful beneficial effect for understanding our animals and plants, and therefore for conserving them, as you'll see in a little while. So I'm not going to go through these plates in great detail and tell you what they all are right now, because that could take the rest of our time, and point out all their characteristics. But I think as you scan your eyes over them, you'll see that the males on the left, the females on the right, the different subspecies up and down have different sets of characteristics: brighter brown, more purple reflection, dark on top for females, light on top for females, the amount of white in that outer band, the amount of dark in the central submesial band and the anal projection of it in particular. The width of the margin. Does the male have a narrow back margin or a broad back margin? Here is the subspecies named iridescens that has strong purple reflections. The California subspecies has no purple reflections. And so you see there's a great array of these kinds of characteristics. And we summarize those in a chart where you can simply go from one to the next and see which trait we're looking at to separate these out from one another. We didn't just do it on one or two butterflies. That's why you've got to have a lot of them. With 6,000 specimens, we were able to make samples that were statistically significant, and our statistics bore that out. Furthermore, we were able to look at enough of them to know that we were within the margins of variation. We weren't simply naming outliers, which has been done a lot in the past with subspecies, which has done nothing but to clutter Pelham's catalog with synonyms. Subspecies are a great responsibility, and certainly species too. And one should not name new ones without being pretty darn sure you know what you're doing. Now, I want to say at this point that the paper that we wrote, whoops, some of you have it. It was in the Journal of the Lepidopterist Society, printed as a reprint. Paul and I took some of them to the meetings in Corvallis, but it's available from me as a PDF very, very easily. And so if any of you would like to study the actual differences among the subspecies and to read the facts about their fascinating biogeography and so on, and to see the pictures, just email me and I'll send you a PDF forthwith. Now let's get back to the story. Much of what I'm going to talk about is history and people and discovery and so on. Then we'll get back to the butterflies themselves, their life histories, their habitats, their evolutionary history and biogeography, and their conservation. So back to macaw. Even when we were calling this charlatensis, way back in the late 70s, I was calling this butterfly the macaw copper. And that was very important for conservation purposes, that it have its own name. And we got a lot of attention to the butterfly and some habitat protected because it had a name. Now, if I had not called it macaw copper, I might have named it Sally's copper. And we named some of the others after women in Lepidoptera, and it would have been very appropriate. In some ways, I was sad not to do so, because she caught the first one, kicked off this whole thing. However, the macaw Indians are the people who have traditionally occupied and still occupy the habitat where this butterfly lives. They're co-inhabitants of this butterfly, and they absolutely deserve the initial impulse to name the butterfly for them, which I did, by the way, only after gaining the permission of the macaws to do so. You cannot assume, it's a presumption to assume that they will want the butterfly named for them, because perhaps they'd see the act of naming as an act of cultural appropriation or scientific imperialism. So I feel the same is true whenever, John and Caitlin, we name a new taxon after a Native American group or their land, that we should probably consult with the people first. And I did with the tribal council, and did receive their hearty approval to name the butterfly that. Here's the plant I'm talking about. What is that? That's Sanguisorbus. Sanguisorba, not sorbus, Sanguisorba, which is known as burnet, right? It finally came. Yeah, burnet is a very important nectar plant for this butterfly on Ostram's Prairie. And you can see just how dramatic that white frosting can be on that butterfly. And note that this mark here, the discal mark, doesn't go all the way to the marginal spots or even come very close. That's a pretty important trait. So that's the macaw copper, the first one. Well, we noticed then, not too many years after that, that these butterflies were occurring south of Ozette. Elsewhere in the Hoh River drainage, John Fleckenstein did a study of the macaw copper for the state and found sites and colonies south of the Hoh River and on down into the Quinault. I found the first colony of this butterfly that we knew about in these bogs, which are the Quinault. They're known as the North Bay bogs, the North Bay Natural Area Preserve of DNR. And they occur between ocean shores and Willapa Bay, the northern part of Willapa Bay. And they, do I mean that? No, I mean Grays Harbor, of course, between Ocean Shores and Grays Harbor and the mouth of the Hump Tulips River. Quite a narrow area that we thought that butterfly was restricted to. But then we found that the butterfly actually occurs in a patchy way between the Ho River and really the Hump Tulips River. And when we examined that butterfly, we found that this disco mark actually extends all the way out to the outer spots or very close to it. And there are a number of other traits that are different too. It has more duskiness in that outer band, and it doesn't have the white orbs. So this butterfly became viewed as a new subspecies too. And we named it after, oh, here it is. I'm sorry, here's where it occurs. Up to the north of here. I don't have its northern range on this map, but what we found was, that is what Mike Patterson found, who's with us right now. He's the one with the mustache.
That's a really old picture.
Yeah, but Mike, it's a really cute picture. Yeah, the guy isn't bad either. But Mike discovered the butterfly in quite recent years, and Kathleen Sayce helped him to flesh it out here on the Clatsop Peninsula, which is very close to where I live. I live right where do I live? I live about here. No, I don't. I live here, right here. And it was absolutely disgusting to me that for many, many years I lived very close to a new range extension of this butterfly, and I never found it, but Mike did, and Kathleen helped him to flesh it out. We did, however, have an old record. It's our oldest specimen from Washington, and we do have the specimen. It's from WSU, that we've got it on loan at OSU, and it was caught in 1918 at Ilwaco. Now I'm going to come back to the population on the Long Beach Peninsula toward the very end of the talk. Suffice it to say now, despite Kathleen's efforts and others to look a lot, I've looked too, we do not have any recent records of it on the Long Beach Peninsula. So I assume that the butterfly didn't get any farther south than Grays Harbor until Mike found it down here. And again, I'm going to speculate toward the end of the talk on how it could have got there from here. But just know for now that it does. There's cute Mike. And here is cute June. June Preston, whom some of you know very well. She passed just a very few years ago. And her husband of 70 years or something like that, Floyd Preston, passed just a couple of months ago. Floyd and June were two of the most accomplished lepidopterists in the country. Amateurs who went all over the country in a camper that they called Preston's Portable Lover's Pad and Lepidopterist Lab or something like that. And they went all over the country collecting voucher specimens of every subspecies in the country that they could in order to flesh out the very important national collections at the McGuire Center, the U.S. National Collection, the American Museum, and others. They made immeasurable contributions to the study of North American butterflies and their conservation. And as a consequence of the fact that I took Floyd and June to see this butterfly at North Bay Bogs on their last trip to Washington, I think, we named the butterfly for June. So it is called Thracilia mariposa junia. And here's a lovely female that Mike photographed in Clatsop. Notice that the females can have quite a bit of white scaling on the top of the wings, which is very bizarre. Very distinct trait. Sometimes occurs in macaw, but not uncommonly in Junia. Oh, by the way, if we'd had a little more information, and maybe we'll get there sometime, maybe there are significant differences between Junia and the Clatsop population, and you can bet that Mike Patterson will be a strong contender for its patronym if that should occur. you can fight it out with the clatsips. And there's the male. And notice on this subspecies that the purplish reflections can be quite bright, almost as bright as they can be on Hyloides, the purplish copper, whereas those are absent on Mariposa, and I think on Mariposa penrosi, can't remember. What's this? Where are we? Oh, okay.
You're a Gerhardt bog.
This is Gerhardt Bog? No, it's not.
Yes, it is.
This picture is Gerhardt Bog?
Pretty sure.
Oh. Okay, we'll pretend it's the Siskiyou Mountains of Oregon. Okay, everybody pretend. That we're going down to the Siskiyou.
I could be wrong.
Well, I'll take your word. Anyway, we're moving down the coast now. And we knew for some years that there's a population down in the bogs, different kind of bogs, in the mountains of the Siskiyous, way in the southwest corner of Oregon. And this butterfly had been found and studied by a woman named Mary Petzel, who was a very, very active naturalist, botanist, lepidopterist, student of wasps, and also an artist and conservationist who was a very dominant figure in the natural history community of Southwest Oregon. This one I know is not at Gerhardt Bogs. This is, I know, one of those Siskiyou coppers photographed maybe near $8 Mountain, I'm not sure, but on the spathe of one of the cobra lilies that is characteristic of the bogs down in the Siskiyous, the serpentine bogs. And so Mary had worked with the Forest Service to get the most important bog for this butterfly protected. It was going to be logged all around it. And because of Mary Petzel's work and Lee Webb, her colleague in the Forest Service, and her great friend, they managed to get that habitat saved. And so there was no question when we came to name this subspecies, which has its own distinctive characteristics, as you see. We named it Mary's copper, Tharsalia Mariposa Marya, Marya, M-A-R-Y-A. And this is her book, The Spirit of the Siskiyous, which pictures the copper right on the cover. Wonderful woman who passed a few years ago as well. And then a big surprise came along, a really big surprise. I would say even a bigger surprise than Mike finding the copper in Clatsop County. When a couple of counties south of there, at the Lost Prairie, what does that stand for? It's a Bureau of Land Management, BLM category, area of something ecological concern, I think, isn't it? And so anyway, it's a BLM conservation area in the middle of logging lands. Lo and behold, there's a bog, and it was being investigated by a staff member of the Xerces Society. Here's the bog. You can imagine that such a bog in such a huge forested matrix, long way away from any other known significant bogs, might have something interesting. And lo and behold, the Xerces scientist found Mariposa coppers in that bog. I'm not sure this is that subspecies, but it's standing in for it. And that was an enormous surprise. And not only that, but it turns out to be one of the most distinctive of all the subspecies. Well, it's one of the most isolated. So one can imagine it's been isolated for quite a while and very possibly has undergone a good deal of microevolution in situ. And so it became the next taxon to discriminate and to name, and we named it for the Xerces employee. I wish I had a clearer picture of her wonderful face. This is Of course, we always do this, don't we? I know very well. Who it is Candace. That's why I'm confused, because we named the butterfly Candia for her, Candia, but her name is Candace. And that's what caught me for a minute. But her last name is Fallon. Candace Fallon is a Xerces Society butterfly ecologist. And can you imagine how happy that makes me feel when I was hoping from the very beginning, 50 years ago next month, December 9th, that's when Xerces started. And from the very beginning, I hoped that Xerces would be a conservation presence, yes, and a social presence and a butterfly study presence, but would also be a scientific influence, and that we would actually have a scientific input to what we know. John Pelham got his video, good. And we do. So for a Xerces scientist to go forth into the world and find a new automatically endangered subspecies of butterfly, one of the most narrowly endemic taxa of butterflies in the entire country, well, that makes me just happy as a cat. But that's not the end of the coastal discoveries. For some years, Art Shapiro and others who have studied the Trinity Alps and the other mountains of the Siskiyou-Klamath region, the so-called Klamath Knot, a very tangled geologic setting in Southwest Oregon and Northwest California. They've known that there was a butterfly there that was the copper, and they just kind of assumed, well, it's kind of an outlier of the Mariposa, the type subspecies, but it didn't really seem like it. And a fellow named Rob Furnau went up into the Marble Mountain Wilderness and studied it a lot and came back with the coppers. He said, these coppers are kind of different. They're kind of unique. They're kind of big. They're pretty dramatic in their appearance. And so Paul, who was doing most of our analysis of the many individuals back and forth, got looking at them. And it was a very fun and fecund period. Every few days I'd get an email from Paul and he'd say, I think we got another subspecies. Pelham's never going to believe it. We got so many. And then we got another one. But dang it, they were all different. And they were all consistently different. And that was cool. This one looks rather like Mariposa. In that it's sandy and buffy below. And I don't think it has the purple reflections. It has a darker central area and it's bigger. It's statistically larger per individual on average than any of the others. Those are not the only characteristics, but those are some of them. And so we decided that was distinct too. And we could have named it for Rob for now, but actually it had been known for a while. And we decided that we were enjoying this trend of naming these butterflies after women who had important contributions to Western lepidopterology, all the way from the last few months in terms of, in Candace's case, to 100 years ago and more, in the case of the person we honored for this butterfly. And that was a woman by the name of Ximena McGlashan, seen here with her sugar daddy, who was her daddy, her father, who was a wealthy banker in Truckee, California. And he had a round tower in Truckee that leaned actually and was built on top of a big rock. It's not there anymore, but you can find pictures of the old Truckee tower. Well, I learned about Ximena when I was a student at Yale, and I was going through all the Lepidoptera literature in the library at the Osborne Zoology Library. And I found this funky old journal called The Butterfly Farmer that was edited by this young woman, Ximena McGlashan of Truckee, California. It's called a comprehensive correspondence course in entomology, conducted under the auspices of the Agassiz Association. Will be a leading feature during the present year. Anyway, that's 1913, if I'm reading that correctly. And so I discovered that Ximena was still alive. This is 1975 or so. And I found her, I tracked her down in Berkeley and I wrote to her. And darn it, I never got to meet her. She was reluctant to come into public. And we didn't get her to come to any meetings or anything. But I did correspond with her, and I have some lovely letters from her in beautiful handwriting in the archives, as well as a complete set of her journal, which I submitted to a major Lepidoptera literature archive run by James Weicker. And she sent me the whole set. So that's Ximena. And she was one of the first women on the West Coast to make a major collection of butterflies, to study the local fauna, to speak about their conservation and their ecology, and to encourage a great many other people through her correspondence course and her journal, to study butterflies. There's even a butterfly named for her. This is, I don't know, John was the name for her or her father. Her father collected a bit too. Anyway, it's Euphydrius Anikia McGlashanai, right? Do I have that right, John? I think John's nodding.
No, it's a Chalcedona.
Oh, thanks. You're right, of course.
And I think it was named for both of them, even though it should have been McGlashanorum, but hey.
There you go. Well, all of ours should probably be orums also, but we went with the feminine suffixes instead. That's right. It's Vesii, named for another important West Coast female lepidopterist, Agnes Vesey, formerly of Portland, who was the first to ever collect the Oregon silver spot on the Long Beach Peninsula, Kathleen. And Agnes Vesey has a checker spot too. That one is Anikia, right, John? Euphydrius Anikia Vesei. But this one is Euphydrius Chalcedona McGlashanai. So that became Tharsalia Mariposa Ximena. And now we're going to move into the Cascades proper, because the Cascadian coppers really weren't very well described by the Sierra Nevada copper. And in fact, there's a distribution gap between them. And they're gray, they're not, but they're kind of an even gray. They're not sandy like the California ones. I think they have some purple reflection. So the fairly extensive Cascadian population deserved a name. Was crying out for a name. We didn't have one all this time for Oregon and Washington Cascades. And so it became known as Cascadia. Paul felt we should name that Tharsalia Mariposa Cascadia, and so we did. Now there's an interesting thing about Cascadia. I'm going to show you the ranges of all of them in a minute, but let's talk about Cascadia for a minute, because it occurs up and down the Oregon and Washington Cascades, almost to the top, but not all the way down. And in fact, there is quite a big gap between that and Mariposa. But the other interesting thing about Cascadia is that it also occurs over here in the Olympics. And you would think that with several endemic subspecies in the Olympics, like the Valorata Arctic and the Spangled Blue and others, that it might be a different subspecies. But no, it doesn't have any major trait differences. So like the Rainier fritillary, the Boloria Chariclea rainieri, and also like the Vidler's alpine, the Rebia vidleri, this taxon is shared between the Cascades and the Olympics. And of course, that gets into our glacial history, which I'll come to in a few minutes now. But we wonder, could there be any modern connection between them? And in recent years, we've had some great discoveries that haven't really quite linked them up across the bottom, but it has brought the two taxa much closer together. First of all, Bill Yake and perhaps others found Mariposa coppers down here in the Southern Olympics, quite near the bottom, as have Tana and Woody now since. And then Mary McCollum, actually earlier than that, found a population way down here near Shelton in the bottom of the Kitsap Peninsula area around the Skokomish River. Quite out of place from the mountains. And since then, Tana and Woody Knaus have found a colony even closer to Puget Sound. And it has some very weird things about it. So this is all very exciting. You see the exploration goes on and on and on. And because of the human disturbance all across here, we're not likely to find any colonies through here, but we may find some much farther down from the mountains to ultimately show how that link works. Who knows? Maybe it's even in the Black Hills. Somebody should go check the Black Mountains and see if it's there. Okay, to carry on then. Just a couple more subspecies. Well, if you look at a female of the subspecies Cascadia, they're usually quite bright orange above, like some of the others. But if you get a little farther east and a little farther north, not only north, but really east, because even in the Wallowas, and from the Wallowas up into the Pend Oreille, and then westward a bit, you get a very, very dark subspecies. Not every individual, and they're not all this dark, but by gum, they can be black. They can look like males. Males with more black on them than males usually have, with just a tiny bit of orange, and sometimes no orange at all, practically black. The first time I ever saw that, I was up in the Okanagan in the Lone Frank Pass general area where we go for the Freya fritillary. And Tia and I were sampling, and Tia came back and she showed me this little black butterfly. And she said, "What the heck is this?" And I said, "Well, I guess it's a male Mariposa Copper." And then I said, "That's no male, that's a female." I had never seen those black females before, even darker than this. And so suffice it to say that when this became a needful subspecies to name, there was no question in my mind, and Paul happily agreed and readily agreed, that it should be named for my late wife, Tia. And that is she. And she would have liked to know that. This is how she travels around with me these days. She's got the cutest driver's license probably in history. And this is Tia Linnea in a patch of Linnea. So Tia's copper, Tharsalia, Mariposa, Tia. Well, then it turns out that there's a subspecies to the north and the east of there that covers just a vast area of southwestern Canada. Even gets out into some of the little mountain ranges. I'll show you in a minute on the map. And just covers a heck of a lot of the southeastern part of BC and over into Alberta and so on. So for us, there was no, and it has very distinctive characteristics. So there was no question really that should be named to commemorate the great work of our Canadian colleagues. And who better to choose for a patronym for that than one of the chiefest among them, who is Felix Sperling, great geneticist, great lepidopterist, great friend of many of us. So this is not it. This is another Junior, but oh wait, that's for the next subspecies actually. I'll just say. So this became Felix's copper or Sperling's copper. And you know, I don't know if this is sexist or not. We used the first names of the women whom we commemorated, and we could have said Felixorum, but we didn't. We chose Sperlingi for his patronym, and so it's Tharsalia Mariposa Sperlingi. And more about Felix in a few minutes, because he's going to remain important to the story. Now we come to the final subspecies we named. And we're coming back west again into the northern part of the Washington Cascades and the western part of the British Columbia Cascades with a butterfly that is particular, and this is another Junia from Clatsop Bogs, but I show it because it's such a good example of the purple reflection. And this subspecies is very dramatic in its purple reflections, although the underside is really quite a bit like Cascadia and Tea. But because of its deep purple reflections and some other traits, it became a new subspecies too. Tharsalia mariposa iridescens. Paul felt we should name it. You could say it's for the Greek goddess Iris, the goddess of the rainbow, or you could say it's a descriptive name, iridescens, because it iridesces so brightly purple. And here are our honorees. Xymena McGlashan, June Preston in action, Felix Sperling, Candace Fallon, Mary Petzel, and Tia Linnea Pyle. So that's our history. I hope you find it interesting. I find the names of butterflies interesting because they tie us into the history of how we know about these things and our connection to other people. So I hope you don't feel that was too much time taken on the people involved in these studies. I hope I talked about the butterflies enough too. But now we're going to hone down toward the end here on the distribution and the biogeography and the ecology. Here are then our now 12 subspecies, what used to be 3, our 12 subspecies of the Mariposa copper. Look at that for a minute. And allow me to say, excuse me, that this paper and the study as a whole is infinitely the better for this map that was prepared for us by, you probably know who, Caitlin Labar, who is, of course, co-authoring with John Pelham a great new study of mapping of all of the Western butterflies. She's a genius of a cartographer, as well as a wonderful and very strong field lepidopterist. So, Caitlin, thank you for this, because we got to see the picture of how they all look on the landscape, right? So here's number 1, the type subspecies Mariposa mariposa, and you see it's actually a little bit restricted in where it occurs in the Sierra Nevada. Number 2, the Marble Mountains population in the Trinity Alps. Xymena. Number 3, Marii in the serpentine areas of the Siskiyou. Number 4, Cascadia from about the the Cascade Siskiyou National Monument on up into the North Cascades National Park. More or less continuous, but not quite. There is the Columbia River and a lot of dry lands that it doesn't penetrate, but does come around here into the Olympics. Number 5, this is Tea, actually extends on down into the Western Rockies. That's quite a large range beyond Washington. In fact, it turns out that Penrose eye is really one of the guild of butterflies more or less restricted to the Yellowstone, the greater Yellowstone ecosystem, along with Beloria crimhild and Hayden's ringlet, the crimhild fritillary and the Hayden's ringlet, Cononypha haydeni. Butterflies that are virtually endemic, restricted to the greater Yellowstone ecosystem. Tia hems it in on the east. And then there's an interesting outlying population in the Black Hills of Penrose eye. Though on further study, who knows, it might turn out to be a worthwhile subspecies too, but we did not have much material and we did not pursue that. And then you come up here and abutting those two with some biogeographical barriers, really pretty much the crest of the Rockies to the east, and the east of the trough, Great Columbia Trough, is this wide-ranging subspecies all the way up into the Northwest Territories, and that's Sperling Eye, which abuts to the west this brilliantly colored purple subspecies that also abuts Cascadia on the south, called iridescens. Now, this might seem very strange that the border runs along the border, because as we know, the borders are artificial, but not entirely so. Actually, iridescens traits penetrate Cascadia traits, of course, and we know there's going to be some interchange of traits on these borders, naturally, for all of them. But interestingly enough, this border is somewhat real in nature. When you go up into these northernmost mountains of Washington and Ferry and Ponderay County and just to the west, you're looking down into a deep trench that's really very dry. You don't get more high mountains right across the border through here. You do over here, but not here. And so this border is actually somewhat real, not merely imaginary. And this border has some reality to it. It's the border between iridescens and charlotensis. So charlotensis for a long time was thought to be endemic to the Haida Gwaii, but now we know that it actually occurs way up into Southeast Alaska and down through the islands and down through the coastal troughs and the fjord lands of British Columbia and Southeast Alaska. It doesn't cross the summit, and nor does iridescens. They really have a bit of a zone in between where you probably can't find either one of them. So how in the world did Haida Gwaii connect with the mainland? Well, we're going to come to the zoogeography here in just a minute. So now you got a picture of the ranges of this entire butterfly. And in Washington, we have the highest diversity of all of this butterfly. Isn't that cool? Well, do we? Let's see. Oh gosh, I didn't even mention macaw. I'm not finishing with charlotensis. Here's macaw, and here is Junia down into Oregon, and here is Candia. So as you see, Junia and Candia are both extremely narrowly confined in the world, these spots being much bigger than their actual habitat. And in fact, those are two of the most narrowly restricted taxa of North American butterflies, and probably two of the most highly endangered, which I should hope enables you to see the power, the descriptive and prescriptive power of studying a vast array of these butterflies and the many, many specimens that our generations of field workers have bequeathed to us. Without those 6,000 species for specimens, folks, we would have no idea. Oh, excuse me, how did that happen? That we have these two extremely endangered taxa. So we thank the specimens and their collectors, as well as all the watchers and students. So how did all this happen? Well, it's complicated, and of course it's conjectural. I want you to get our PDF and read the description of our hypotheses for their movement over the years. But you got to realize that it all has a great deal to do with glaciation. And when the Wisconsin advances occurred, we got the Cordilleran ice sheet down over much of northern Washington, down into Puget Sound, down into here. And this shoved the butterflies around. Was Haida Gwaii perhaps a refugium? Were there any other refugiums up in here for Mariposa coppers? We don't know. There might have been. There might have been surviving coppers to the north on the western fringe, There certainly were in the south, but the ice pushed the butterflies around, and the Arctic alpine conditions extended well beyond the paraglacier edge, the edges of the glaciers. And so consequently, there was probably a lot more distribution down into the Rocky Mountains, possibly farther down into the Sierra Nevada than there is today. It's probably retreated back since then. But you cannot begin to picture how the distribution of this butterfly came about without remembering this massive advance of ice. Oops, wrong way. So here we have the Wisconsin Cordilleran ice sheet advance into Washington. And we may have had a refugium of the Coppers in the Olympics, in BC, and in the lower edges of the Cascades. But we don't know if they were already there beforehand or not, or if they emigrated into this new habitat when the ice retreated. Because when the ice retreated, it left a lot of peatlands, a lot of bogs and marshes, where the kettles of ice remain. When they begin to melt, you get moraines, of course, but you also get kettles, which are these low spots where marshes will form, or not really marshes, but bogs. And in those bogs, you get the array of food plants that these butterflies use, which we'll get to in a moment. So when we look at the modern configuration of the Western states and provinces, this is what we have now. And the green indicates, of course, not only the forest, but the more mesic areas, the moister areas. You're not going to find the Mariposa copper in the brown areas. It doesn't occur in the drier areas. It occurs in moister forests and bogs and those kinds of places. So down in here, down in here, down in here, down in here. But where did it go to get into these places? I'm actually going to read you a paragraph rather than try to summarize it. It'll be all over much sooner if I do this, and it'll also be clearer as we wind on down. Let's see. Is that the slide I want? Let's use that one instead. This is actually a butterfly called the bog copper of Eastern Canada and the northeastern US. You can get it in Wisconsin and Minnesota, all the way east into Maine, and then up into the Maritimes and westerly a ways in Canada. Different species, but clearly closely related. And it might come into this picture. So here's a summary of how these butterfly distributions may have come about. Overview and origins, excuse me. In summary, we suggest that the distribution of Lycaena, now Tharsalia, Mariposa, can be interpreted as a ring species. Actually, to do this, let me go back to the range map for a minute. A ring species, a classic pattern with the possibly very old ancestral Rocky Mountains. No, my finger doesn't work. Possibly ancestral Rocky Mountains. Penrosea, the southeastern extreme, extending northerly through Sperling Eye on the east side, and Tia on the west side, and Iridescens on the west side of the Continental Divide. Then ringing back down the outer coastal reaches through Charlotensis in the north, followed by Macaw, Junia, Candia, and Maria, progressively Ximena, Ximena, and finally down to progressively and finally down to Mariposa at the southwestern extreme. Any connection that might have been born between Lycaena mariposa and Lycaena mariposa penrosi from here to here across the southern mountains has since been lost or precluded by post-glacial warming and drying and imposition of the Great Basin and the Red Desert, south of which no Mariposa coppers survive in relictual boreal outposts. So when it was cold, the coppers might have been all the way across here, and they might have had their origins here or here. We don't know. Alternatively, the ancestry of this species may lie in the boreal northwest with, in the boreal northeast with, with subsequent dispersion west and south. Lycaena mariposa is in many ways and places an ecological cognate species. Do you know that concept of cognate species? Species that are closely related and they fill the same niche, but in very different places. And in many ways, it's a cognate species to the bog copper, Lycaena epixanthe, in the Northeast, whether by habitat convergence or descent is not known. So this butterfly might have gotten there from an ancestor of both the species, or it's conceivable that Mariposa could even be the ancestor species of epixanthe. Other possibilities include a Sierra Nevada endemic origin with dispersal northwesterly and northeasterly during a wetter time following the glacial retreats, or Beringian beginnings from, and that, you know what that means? Beringian means coming across from Europe, where there are species just as purple as this. And I don't mean Europe, I mean Asia. Eurasia is what I meant to say, but we're really talking about Siberia and northern Eurasia. There are species that look very much like epixanthe, just as purple. And it's possible that both Mariposa and epixanthe, the bog copper, had their origins, had Beringian beginnings from some whole Arctic, that is old world ancestor that came across the land bridges. In the absence of a phylogeny revealing nearest relatives, any of these scenarios might be equally plausible. What is certain, and here's your take home, what is certain is that multiple successive glacial stadia and interstadia, volcanism, fire, drying, forestry, and other landscape scale changes have enabled mixing, isolation, and remixing again and again, and continue to do so. Figure 3, the range map, represents our very best effort at parsimony in ordering today's complex evolutionary riddle. And so you see, this butterfly has really been bashed about by many, many forces, and that has caused it to settle down in certain places where it's been left alone long enough to undergo local evolution under the local conditions, and thereby to acquire this great array of variation that it was our privilege to discern and to share with you. Butterfly itself, as you know, is always two different kinds of animals. And in the case of the Mariposa copper, it's a classic, people say slug-like, sort of slug-like, sort of pill bug-like, lycaenid larva. This is what they look like. And they feed on an array, as far as we know, of vacciniums. That is to say, huckleberries and blueberries and cranberries. Now, the macaw copper that we first found up in Ozette Prairie is in the bogs, as is the junior copper. We could call it the Clatsop copper, couldn't we? That'd be nice. It'd be a nice common name, Clatsop copper, June's copper, Clatsop copper. Take your pick. It's your choice. Glass of copper has a nice ring. June wouldn't mind. But that's in the bog too. And so you see sphagnum moss, you see sundews. I think there's sundews in this picture, aren't there? I thought there were. Maybe not, but they would be there, as well as the bog cranberry. And the bog cranberry is the host plant of the macaw and June's coppers. Now, it's interesting that the rare habitat limited subspecies, your macaw, your Junia, your Candia, your Marii, and to a lesser extent, your Ximena, occur on the boggy, more mesic wet country cranberries and possibly huckleberries. But when you get up into the mountains and you get your much more common coppers, the Sierra Nevada one actually is kind of in between. It's not very common, and it's in pretty wet areas in the Sierras. So it's kind of transitional. But when you get into your Rocky Mountain coppers and your Cascade coppers and your Sperlings and your purples and so on, then you're getting, and your Tias coppers, you've got common butterflies, not endangered, live over great areas of habitat, and they feed on much more xeric species of huckleberries. That is, ones that can grow in drier conditions. They don't need bogs. They can occur in bogs. And so when you go to the edge of those nice big wet meadows, like McClellan Meadows or Government Meadows up in the Cascades, yeah, you find coppers in the boggy parts. Sure, they'll do it. They'll feed on the boggy cranberries, but they'll also go outside and feed on cranberries like this, which is Vaccinium cespitosa. The bog cranberry is Vaccinium oxycoccus, and it'll use them, but it doesn't need them because the common subspecies can feed on Uliginosum, and it can feed on Deliciosa, the ones we like to feed on, and even ovadum, the cranberries that grow up on bushes. They can even use those. So we share host plants when we have the chance with Mariposa coppers. But something really weird has come up lately, and that is that I don't know if Woody and Tana are with us tonight, but Woody and Tana Canals over in Kitsap country, Hoodsport, have been finding all kinds of cool butterfly records in the Southern Olympics, as has Bill Yake. And they've also found Mariposa coppers on a power line right away in Mason County, getting perilously close toward Puget Sound country, giving us this great link of Cascadia across the bottom. Well, where they found them was among rose spirea, also known as Douglas's spirea or Douglas's hardhack. Very common plant in many of these wetland and wetland edge habitats where we find coppers and other butterflies like that. And it's a wonderful flower. It smells so good. It's so pretty, but it's a crappy nectar plant as far as I can tell. I don't know if I've ever seen a single butterfly nectaring on it, unlike some of the white spireas up in the mountains. But Woody and Tana found the butterfly, they think, ovipositing on this. What's that all about? Well, a couple of possibilities. One would be, of course, that it's actually a host plant. That would be remarkable. I've corresponded recently with hello in California. Tell me, John, if you think of his name, Lycaena biologist who did some of the early work on Mariposas and Vacciniums. You know who I mean. Can't think of his name right now. Anyway, he told me he doesn't know of any food plant records on this. Nobody else does either. So this would be the first record of Mariposa feeding on a rose family plant that we ever had. It's not just called rose spirea, but it is a rosaceous plant. It's in the rose family. So that would just be weird. But the other thing it could be doing, when the bogs have this plant, they're pretty darn thick. And it might be hard for the butterflies to get down to the cranberries. You've seen this, Mike, at at the class of bogs, it's easier to find the coppers where you don't have a lot of Labrador tea or spirea. But that doesn't necessarily mean the butterflies don't use it. Maybe they're laying on the shrubs and the caterpillars are crawling down and finding the cranberries, the bog cranberries. That's a possibility too. So more to learn, folks. Keep your eyes out. You might be the person to make another great discovery for these butterflies. Now, how about when it comes to cranberries? Mike Patterson made a cool discovery, which was that another cool discovery of his many. And that is that the class of bogs, which they're working on getting them protected by the North Coast Land Conservancy and others. I guess the key habitats aren't quite fully protected yet. But anyway, there is protected land there. And it turns out that those bogs on the Clatsop Plains, those back beach, back berm bogs, which are pretty extensive, they're not all in good condition, have not only bog cranberry, but they got commercial cranberries, because many of those bogs had been converted to commercial cranberry bogs, as had almost all the cranberry bogs on the Long Beach Peninsula, which we'll come back to in a minute. Well, a lot of those bogs were abandoned, and some of the Clatsop bogs that have the coppers must have been abandoned commercial cranberry bogs, or at least the commercial cranberries crept into them, because you can now find both the commercial cranberry and the bog cranberry in the same bog. I think we do not yet have any records anywhere of the Mariposa copper feeding as larvae on the commercial cranberry. I have looked in a number of bogs in Grays Harbor County that are not sprayed as much as some others are. We got some good candidate bogs we've been invited to come look at. We haven't had a chance to do that yet. But the commercial bogs are generally under a very strong chemical regime. One time I was interviewing the extension agent in charge of cranberry growing on the Long Beach Peninsula. He came and talked to our grange, and he told us that the chemical rotation on these bogs on the Long Beach Peninsula consisted of 11 chemicals rotated through: insecticides, fungicides, herbicides. And you can imagine they don't have one heck of a lot of insect diversity left after that. And that may have accounted for the extinction of the Mariposa copper on the Long Beach Peninsula. It's a beautiful and wonderful industry, and a very important one where we are. But very little of it is done organically. There are very few organic cranberry farms. It's hard to do because there's a lot of insect competitors, and there are fungal problems as well, and weed problems. That's why they use all those chemicals. And they end up with a terrific crop that I hope doesn't have too many chemical residues. Every time I eat cranberries, I worry about that. But the bogs sure don't have any coppers in them. Now, we may find them yet. June's coppers, here's one of Mike's great females over there, class of bogs. As I told you, used to occur at Ilwaco. There was one record, we don't know where in Ilwaco, but we suspect it was probably down near Cape Disappointment in the marshy areas down there. There aren't any actual cranberry bogs down there yet, but Kathleen Sayce has determined that up in the middle of the peninsula, there are some remnant bogs that have never been converted. I know there are wetlands up there. There's Lake Loomis, which is actually where I rediscovered the Oregon silver spot on the peninsula in '75 before it became extinct in the peninsula. But up in those bogs, if we can ever find a way into them, we may still find that the Mariposa copper occurs. Kathleen, are you on there still? Do you want to say anything about that?
Yeah, sure. I unmuted for a minute. There's rather an extensive area north of Cranberry Road called Island Lake Forest that's now owned by the Columbia Land Trust. And it has at least 2, possibly 3 natural bogs. In undisturbed forests, it's actually native Doug fir forest, which is really cool, on top of having the bogs. But it's got some undisturbed bogs in there that do have the native cranberry in it. They also have gentians and other species, lots of sphagnum, very high quality. The problem that we have is we used to be able to get in there very easily, but the windstorms in 07 and 09 blocked the access roads, and you now have to go in by water, paddle across the lake, and then bushwhack up through the forest and over the hill to get into them. So we just haven't been able to get back in.
Yes, I didn't know it was that the land trust had acquired it. That's wonderful. Well, we're gonna have to recruit some extremely energetic, dare I say, young people to do this, or I don't know, get the land trust to hire us a helicopter one day. We could do that, right?
Yeah, not with those trees.
No, you're right. Absolutely not. But we got to get in there somehow and try to find it, because it would be very cool if the coppers were still in there. Thank you, Kathleen, and thanks for all you've done for this and the other butterflies, silver spot, and other things over the years. Mike, do you have any comments on the classic animals? Mike Patterson, you still there? I'll come back and see if he wants to say anything later. Meanwhile, let's ask the question for a minute. How in the
I
world?
think I was on mute.
Okay, Mike, you got anything you want to say about classic? Copper?
Well, as you've been into the site where the coppers are at Gerhard Bog,
Yes.
and it isn't actually in the protected area. It's in an area to the north of that. And this last year, somebody put up no trespassing signs.
Oh, no.
Yeah, and so we don't know what the new status is, or whether the North Coast Land Conservancy has an easement into that, because you can access the North Coast Land Conservancy from that side. So I don't know there's an easement there. We don't know the circumstances for why it was that sign was put up there, whether it was the people who own the property that nobody's been able to get in touch with, or the folks who have the easement up to the communication towers, or that really angry man that you and I ran into when we visited.
That's
We
what I was
just
going
don't
to ask.
know.
That's what I was going to ask. Was it that guy? And Mike, you're blameless in this. I managed to really tick off this guy by parking too close to his car and driveway. And actually, I parked pretty badly, and then I kind of mouthed off to him too. It wasn't very nice, and he got really mad at me. And Mike suffered for it. So if the butterfly becomes extinct before the tsunami, it might be my fault. Thank you, Mike.
Well, we're still looking in the parts of Gerhard Bog that we have control over too.
Indeed.
And there's another spot up near Cullaby Lake that hasn't really been surveyed very hard at the right time of year. So we got some other work to do.
Great. And those classic bogs are extensive, lots of different conditions, I know, but I'd be surprised if there weren't more populations than you've been able to get to discern. So that's exciting to look forward to that. And I'll try to be nicer to that guy if I ever see him again. Let's talk for a minute about how Junior could have possibly gotten to Clatsop or vice versa. So we know that it occurs, it occurs from Macaws here. Junior picks up south of the Hoh River. So Junior occurs basically through this strip right here, not continuously by any means. There are only a few known populations. And then there's the old record right here at Ilwaco from 1918. And then, I'm sorry, here at Ilwaco. That was Tokeland, here at Ilwaco. And then down here in Clatsop. So how in the world did it cross? Well, remember in the glaciation, it wasn't just a matter of the glaciation, the glacial advance and maxima, but it was also the flooding. So here's glacial Lake Missoula, the Clark Fork Dam near Missoula, and the great inundations that occurred time and time and time again with the melting of the dam, known as the Missoula floods, or these days as the Bretz floods, after J. Harlan Bretz, who hypothesized the fact that these floods had occurred, and nobody believed him. He was an iconoclast, and so someone asked him toward the end of his life if he was happy now that he'd been proved right that the Bretz floods really occurred, and he said, "Well, yeah, but I don't, the savors got out of it because all those guys are dead now." So they weren't there he wasn't there to gloat over them. They weren't there for him to gloat. Anyway, this became the biggest flood that we know of. And I've got a song poem about it called Brett's Floods. And one of the lines is 10 times the flow of all the rivers in the world. And that's true. When the Brett's floods were in full flood, it was, 10 times the flood of all the rivers in the world. And it carved out the channel Scablands, Wallula Gap, all the rest of it. And the current maw of the Columbia as we know it. Prior to this, it's entirely possible that the mouth of the Columbia was a braided affair, no gorge at all, but more like the Mackenzie or something like that, with occasional connections across the changing course of the river. So it may have been entirely breachable. And there are some other species. There are some species that do not span it, like certain salamanders. I think Dunn salamander and others. But then there are others that do. And we can only assume that their movements occurred at different times with different glacial history and before and after the floods. And so that's what I have to say. And I would like to conclude with a little pastiche. I don't mean that. I mean a little montage of the Clatsic copper, of June's copper. These are mostly Mike's pictures of this very beautiful butterfly, just to leave the pictures in mind, and then we'll go to Q&A. And I'm sorry I didn't break for that earlier. I love that butterfly with that leaf, Mike. There's a pair. And a purpley male with a purpley leaf. Such a butterfly. Thank you very much for listening. And let me just give my acknowledgements. Oh, yeah, that's one of my favorites on the Salal leaf. Was that yours, Mike? Or that might have been John Fleckenstein. I'm not sure. Thank you, folks. Thank you for listening. Thank you to all these people. This was a 40-year project. That's why the talk is almost 40 years long. I'm sorry. And first of my acknowledgements, of course, goes to my co-author, Paul Hammond, sine qua non, without whom nothing, but a number of these people were sine qua non. Never would have happened without John or Kate or Sally finding the thing in the first place with my collecting party. Tia out there in the field with me a lot, first poking my nose in the subspecies that came to bear her name, which is one of the reasons I decided, by the way, that John and John were absolutely right. You can't describe, and I wish I'd said this before, you don't go describing one subspecies of a diverse taxon that clearly has a lot of genetic diversity and just describe one out of context. That would have been not only impolite, but rather stupid biologically. It was really when I saw those black female teocoppers that I said, oh yeah, we've got to review this entire species if we're going to do this at all. That's why it happened. Chris Marshall, Dana Ross at OSAC made the pictures possible and the specimens. Folks from various museums in both countries came through for us. The Makah people, Haida, Quinault, Chinook, and Clatsic peoples, everybody else. On those lands, we wish to recognize and thank and honor. A bunch of the agencies took part over the years. John Fleckenstein surveyed them for DNR, and Mary Kelly up on the Quinault Reservation. He's the first to find those very cool white females that can be damn near white up in the middle of the Macaw lands. And he's an enrolled member, by the way, as well as a state biologist. Very, very lucky for us. Janet Berger was the first to find it way up in the North Bogs above the Olympics. Candace's great discovery, Mary's, Lee's, and the rest. Folks down in the Siskiyous, Linda, Rob. And then Mike and Kathleen, I can't thank you enough, Bill, and then Tana and Woody coming up with some of our most recent discoveries, and my field companions, David, Florence, looking after me these years, and all the photographers. And by the way, I pirated pictures from lots of people. Financial support from these sources.
Okay, well, thank you so much, Bob, for a wonderful presentation. I'd like to start by going over some of the comments and questions in chat, and then we can open everything up completely. First of all, Kathleen Safes noticed that the picture that was in dispute about whether it was Gerhardt Bog or the Siskiyous, she noted that there were true firs, and so she infers that it was from the Siskiyous, and excuse me, might confirm that. BLM area.
Thank you.
Area of critical environmental concern, FYI. Let's see, I wanted to put your email in chat so that people can write to you for a PDF. So which address do you want to use?
Yeah, let me do that. Let me call up a chat here.
Okay, you can put it in chat or I can put it in, either one, but I want to make that available to people.
Yes, right. I have a couple of addresses, they're both Tia's old addresses, but please write me with this. Don't send giant files to this address, but please request the PDF from this address, tlpyle@willapabay.org.
Okay, so tlpyle@willapabay.org.
Correct, and I'll send you the PDF right away, happily.
Okay, Data Droppers was querying about the DNA work that's been done. John put in a comment. Maybe we can open that up here in a moment. The other request was to have a look at the larval photo. And Linda Kaplan, it seems like may have a question.
Great. Excuse me.
So could you go back to the larval photo?
Yes, but okay, are we going to do the DNA question first or the caterpillar first?
Let's go to the caterpillar first, because I think that will I think the DNA question will be bigger, and then John. So once we deal with the larval issue, that question maybe that Linda had or comment, then we'll open it up. And I know John Pyle has a comment as well. So Linda, did you want to say something about this or have a question? Feel free to unmute and we can just start to have general discussion.
Excuse me.
Okay, well, if since she's
Linda,
not...
Linda, are you there, dear? Linda, you might be muted.
Yeah. Okay, well, why don't we go to the DNA question, and then, as I say, John Pelham has a question as well. Or
Okay, for the DNA question, is there an actual question, or is it just DNA question mark?
David was asking, go ahead and unmute, David, you can ask the question yourself. He was asking about whether DNA work had been done, and John commented that it's not always helpful for subspecies.
It's you want to say anything, David? Okay, it's a perfect question for my final slide. This is Felix again, obviously. And the reason I brought Felix up for the last slide is to ask the question, what comes next? Now, it's true that DNA is not always adequate for subspecies. However, if it's resolved finely enough, looking at enough sites checking out enough genes, enough parts of the gene pool, then it's possible to get resolution that may be at least suggestive at the subspecies level, particularly if the subspecies have been separated for some time. In fact, there's a point in our paper that I'll maybe come back to that. But actually, there's a real difference between subspecies that have kicked off quite recently and subspecies that haven't, obviously. And then, of course, you come into the whole judgment of where does speciation occur. And in fact, when Paul and I gave this paper, a version of this paper, a little more technical, little less history, at the Northwest Lepidopterist Association meeting, the chairman of the biology department approached us afterwards and he said, looking at your data, I wouldn't be surprised if you've got some species there. Now, I would not contend such a thing, but it's possible. And certainly if that's the case, the DNA ought to be able to help. So what I've done is, I say at the end of the paper that here we go. Let's read this. Of course, what Paul and I did was a traditional, conservative, traditional phonetic analysis. We looked at the actual anatomy of these things, their looks. We checked out their looks the way people have always done. What we did is essentially Aristotelian with some more modern advances. It's what taxonomy always used to be, and it's still done that way. However, we now have the modern tools that Paul and I are not the masters of. So what we said here is that, of course, this, there's some recent work done on the Lycaena xanthoides, the large copper, and on Lycaena edith copper that was genomic. And that, of course, came up with some very important highlights of inferring species boundaries and identification of cryptic lineages. And that paper concluded by saying inferring evolutionary relationships among closely related species should benefit from multiple sources of information. And I say, of course, this is correct. And therefore, we welcome further analysis of the Pleistocene and Holocene processes that we interpret as having led to this picture of Lycaena mariposa's development. And we also welcome molecular work, whether mitochondrial DNA, microsatellite, or single nucleotide polymorphism SMP driven, or any other method to test our systematic conclusions. And we've seen how dramatically last month with Caitlin, how dramatically that work, such as Nick Grisham and others have done, can solve some of these problems, or at least point the arrow. And so one of the most talented Lepidoptera geneticists anywhere is Felix Sperling. And to my enormous delight, after we named the butterfly for him, which he agreed to, he fell in love with these butterflies too. And he said, I'd like nothing more than to do a complete genomic analysis of that species. And so, bravo. And I really, really hope he's going to do so. He might not be able to discern all the subspecies we have, or he might have evidence that allows us to infer that they were correct. That would be lovely. Who knows, he might sink us on some. Hope not, after having got past the Pelham barrier to get them all into the catalog, but he might almost even come up with the idea that one or more are species. So go for it, Felix. Hope it works out. Thank you.
Okay, thank you, Bob. And everybody, feel free to unmute. I see, John, you got your finger up. Go ahead, unmute. Everybody can unmute now, open discussion. We just want to be respectful of everybody's time. So go ahead and open discussion now.
Y'all hear me?
Yep.
All right. A couple of 4 things. First of all, Haida Gwaii is a recognized refugium, and it's likely that it had brothers and sisters up and down the coast. all the way to the Alaska Panhandle. And in accordance with that principle or that idea, the concept, Pacific coastal distribution, I mean, immediately coastal distribution is widely recognized in the number of taxa, including plants and animals, and especially moths and butterflies. So like that whole pattern is like a really wonderful example of repeated evidence. Likewise, the continental divide that you show for the division between Sperling Eye and Tia on the one hand, and Tia and Penrose Eye on the other, is also showing up in our atlas over and over again. It's repeatable. And again, it's, like these aren't boundaries so much as, the biogeographical indicators, just like the Okanagan Valley in Washington is not a complete barrier, but it serves as such for many, many taxa. And the other thing that I wanted to say was iridescence. I came to the point when I was compiling the data for the catalog, and I thought, this iridescence thing is a problem. And that's why it remains in the catalog, because it's a problem that's not solvable by resolution as a synonym in any other taxon. You can't synonymize it to Sperling Eye or Charlotensis or Cascadia. You can't put it any place. It resembles to some degree all of those other taxa, but it remains. And of course, it's probably the last dispersal event. It occupies an area that was totally under ice. Right. So it represents something that's unique and interesting, and so it maintains that name. And last, genomic studies now have, we've left mitochondrial DNA in the dirt. Nobody's, doing that stuff anymore. We're doing total genome. And I think that what Bob said last is really, really important. It's not, a really important thing to discover whether there's, cryptic species or not. It's important to recognize lineages, and we can do lineage analysis with something like Mariposa, and we can determine, to some extent, how long these things have been separated along the coast, or how closely related things are in the Rocky Mountains. And maybe we can actually say, well, there was a Rocky Mountain refugium, we don't know where, but it was, extant. There was a Sierran refugium, and maybe a Cascadian refugium as well. These are important, even if they don't, precisely define taxa or, recognize, cryptic species in there. So that's my piece. I'm done.
Okay, I'd like to respond just a little to that. Thank you, John. That was beautifully put and absolutely spot on, every bit of it. I'll try to do it in the same order you did. I don't have much to say in response, but yes, I was thinking, of course, that's right, that there was a recognized refugium in the Haida Gwaii. And then people ask the question, well, what about how did things get from there to the mainland? It's a deep water trench. Well, there was a much, much lower sea level at the time. And probably the gap, if it even fully occurred at one time, was not so formidable as it is now, just like as with the Columbia River mouth. So it does not beg belief to imagine historic movement from Haida Gwaii to the mainland and back. And wouldn't it be something if the lineage studies ended up showing that Haida Gwaii was the primary origin or source? That's not the word you were using. The lineage generated there from I'm very much hoping that whoever does this is going to be looking at those Siberian taxa too. And we may find that the whole ball game, the whole thing is just it was a big Beringian spread all through the Pleistocene. I kind of don't think so. I think we're probably going to find just that there was an earlier origin with far earlier glacial events, possibly from the Northeast or possibly from Beringia, that then created some rather old populations in the Rockies, in the Sierra, and on the coast. And that we're going to have, what's the word you were using, John, in your last part? You were saying it's much more important than the taxa and names.
Yeah, identifying, relationships among potential cryptic lineages.
Yeah, the lineages. That's right. It's the lineages. We might find that I think there's probably going to turn out to be several older lineages, but maybe not. And then yes, iridescence, it is problematic. It's a glob up there. It's one of the last ones that Paul discerned. And we had to talk ourselves into accepting that one because it's not as, you look on the chart, it's not as easy to discern it as a group of the others, but you look at a big series of them, and that's again where the power of the series of specimens comes in. You can't do this work with a few pictures or a few butterflies. It cannot be done. But then you see the trends. And John, you're absolutely right. I think it's the most recent dispersion event, and it's probably the youngest among these taxa overall. And being youngest, it's going to be the mushier as well. Older, isolated things tend to become just through chance, just through mutation and chance and natural selection, tend to become more discernible, more distinctive than the younger ones. I mean, it's not always true, but it's common. I guess that's my response to what you had to say, John. And yes, we got much more powerful genetic tools now, and thank goodness that Felix has access to all those, and he's interested in doing so. And I hope you'll be able to do for Mariposa what Nick and others are doing for the Celestrina.
Okay, there was a question about genitalia, whether the subspecies can be differentiated through genitalia.
Take it, John.
Well, since I am known for talking about genitalia way too much, we have done some dissections. All of the New World coppers, excluding Cupreus and Phleas, have genitalia that are remarkably alike. I had not done anything that indicated to me that there's any difference between Mariposa and Heloides or any other copper in the New World. So I can't say. I did want to add something that New World coppers are distinct from anything in Eurasia except for Cupreus and Phleas. So like these lineages, all of the things that Bob said are really New World phenomena. And I don't doubt the possibility at all that Eastern coppers may be responsible, or vice versa. Western coppers may be responsible for, like the bog copper in the East. So I'm out.
Now, I did not know that, John. That none of the other whole Arctic coppers have any shared lineage with ours?
No. Well, the Cupreus and Phleas, those are the two.
Just those, which are our most distinctive coppers, really.
Mm-hmm.
I just wanted to say, Bob, that I'm glad you made your PDF available to everybody, because I think it's extremely interesting in showing up a lot of the geological underpinnings of the distributions that you kind of touched on. But I think by reading the paper, you get a much, much better idea of how important the glacial advances and retreats and the floods may have shaped these different butterfly distributions.
Thank you. Yes, there is more to the paper in that. It's by no means definitive, and parts of it, of course, are conjectural. John, conversations with John over really, coming up on 60 years of conversations with John have often come around to biogeography. And John's very much caused me to question more and more and more how these might have come about. So thank you for that, John. For example, when we think about those Haida Gwaii coppers and their close relatives, surprisingly, on the mainland, we realize they might not have been separated more than a few thousand years. And when the glaciers retreated, as I said, a great deal of peatland was created by the pressure on the plants and the leaving behind of great bodies of ice that became water, that became bogs. You got your peat bogs, and the peat bogs, of course, are generative of a whole community of organisms that includes the cranberries and the coppers. So there's a lot of habitat automatically available. Doesn't mean the butterfly is always going to be there, because they're separated. When you go to OZ now, it's quite a little ways to the next habitat of any kind. Well, there's Manny's Prairie. There's a couple of different prairies scattered about. But you realize that 100 years ago, when there was a lot more Native American burning there would have been more of these prairies. Before the Quileute Prairie became airfields and houses and mills, that might have been full of this kind of biology. We don't know that. There's only one other butterfly we've ever found at Ozette Prairie. Never found that fritillary of Lars's. We only found woodland skippers there. It's the only other one. And they're separated from the woodland skippers that are on the edges of the logging roads at Nia Bay and Royal, Washington, and all around there by only a few miles of rainforest, but they're different looking. I mean, I maintain that the woodland skippers of Ozette Prairie are consistently different in appearance from the ones around them by just a few miles. I'm not going to go name a subspecies based on that, but you can get isolation and differentiation on a very minute basis. So when you think about the great big changes that glaciers bring and floods bring and fire brings and warming brings, I mean, right now we're seeing changes to our population, our butterfly fauna, our collection of species, the species we live among, through warming right now. We know we are. We're seeing seasonal changes. We're seeing distributional changes. We're seeing buckeye butterflies coming into the state and so on. We're going to see more. We're also going to see them on a catastrophic basis. If you think that all of these butterflies are safe because the land trusts are coming in and the BLM is recognizing them and Xerxes, well, I hope so. I mean, I could have talked a lot more about the conservation prospects for each of them, but the really big ones, of course, are macaw, Junia, and Candia and Maria. But they might all be in danger from the tsunami. When the tsunami occurs, which will occur, it might be an extinction event for 3 or 4 of these taxa in the world. It could be, or at least dramatically reduce them again. Hope not. They do have a little bit of altitude, some of them. So, but I don't know. We're going to watch and see. So we're in the middle of a big dynamic system here. We as butterfly lovers get to take part in that dynamism. Isn't it fun? And to come together like this every month to hear someone talk about what we're discovering. Everybody come here, Professor Lynn, next month. It's going to be terrific. Any more questions for me now?
Yeah, I have a question, Bob, about local range of the coppers. You know, I'm not saying it because I imagine that, you know, a macaw copper would fly south and merge with the other population. I think it's Junia, but I was just curious if you have any information on that.
Yeah, once again, it kind of comes down into a couple of different guilds of these coppers, if you will. Almost specialists and generalists within the same species. I can't help but look at them that way. These coastal coppers are very habitat specialized. They're extremely patchy because of that. If you look at a book called The Peat Resources of Washington, it's a cool old document. You might be able to find it online. You'll see the extent of peat along the Washington coast. And you can imagine going back 5,000 years before that, it will have been a great deal more. And then back when the sea level was lower and Haida Gwaii was sort of connecting to the mainland, maybe that butterfly wasn't so patchy then. Maybe it was really extensive. But as the marshes, I keep saying marshes, as the bogs contracted and as the plants succeeded in the absence of Indian burning and homesteader burning, and became smaller and smaller and smaller, then these butterflies have become extreme habitat specialists. And I don't think you're going to find colonization events outward from them, unless there's a big effort to create more patchy bogs around them, which, I mean, the substrate occurs, but it would take a huge effort. The National Park Service is having enough trouble just getting funding and getting their fire stuff together to manage Alstrom's Prairie. There are people who want to burn it all, some want to burn it too fast. I mean, it's controversial. You can burn it too fast. As we know, fire ecology is important and controlled burns can be helpful for some populations. But certain rare skippers have been extirpated from Iowa and Illinois because of too aggressive conservation burning by TNC and DNRs. You got to be careful. John and I had to lobby DNR at one time very aggressively to prevent mime mounds from being burned too fast, too hard, and extirpating the rare butterflies there. So no, I'm not expecting to see colonization events outward from these tiny ones, Paulette. The habitat just isn't there. The barriers are too great. But then when you move into the your Cascadia and your Tea and your and to some extent your Penroseae and Mariposa, certainly Sperlingi, you get again these vast habitats that are open to it because these conifers, mid-elevation conifers and higher elevation, have these great hems of Vaccinium cespitosa all around them, like this skirt of these huckleberries occurs everywhere. And you can find Mariposa over vast areas, and they are capable of colonizing. That's why it's really interesting that these subspecies even exist. Why didn't they just totally run into each other? And well, that's because of what John suggests. There are on the landscape some natural, if not barriers, at least inclinations. And that's really what I did, that doctoral study that I was conducting when we found the butterfly that's on the screen right now. I took that pic, that's one of my pictures. I took that picture right there at Alstrom's Prairie. And that's where we first found it. Well, that year, that summer was 75. And the study that came out of it was called the ecogeography of Lepidoptera conservation. And the central part of it was the ecogeography of Washington butterflies. Paulette, I've got a paper from that. It was published in the Xerces journal, Atla, which I'll send you. That goes into the butterfly provinces of Washington a lot more. But in doing that, I realized that there are such a thing as butterfly provinces. There are areas that had different ecological and geological and altogether biogeographical origins. And they are distinguished by different emphases in their faunas. So we have a more Californian butterfly fauna in the Klickitat area. We have a more Central Rockies fauna that evinces itself in the Blue Mountains, and the Northern Rockies fauna that bumps up into the Selkirks and the Pend d'Oreille, and so on and so forth. And these butterfly provinces stand up statistically. Well, the very same thing occurs with butterflies. And that's what John was talking about. Particular species will occupy a big wide area, and then there's something, crest of the Rockies, perhaps a temperature or humidity isobar or gradient that makes it no longer so hospitable for them. And the population may retreat across that barrier into a different headquarters, which will then proceed to acquire over time through mutation, microevolution, distinctive characteristics. That's how this works. But it's also like a puddle in the sun, and you watch a puddle drying up in the sun, and little patches dry out one by one. The ones that are left are called relics, the ones that are separated. And what we have here are relictual populations near the coast. And continental populations on the inland. But then look, on the east side, there are some relictual populations too, in the Cedar Hills and the Black Hills and the Cedar Hills. Those need a lot more examination, really. Those little outliers of 6 and 7, Penroseae and Sperlingi. Might clutter up your catalog a little more, John, when those get looked at carefully.
Oh, I make the ultimate decision.
That's right. That's what we respect and what we love you for and praise you for. And just thank God you made the right decisions for all of these.
I have
tell
to
you-
go. Thanks, Bob. Thank
Hey,
you.
we all better be going. Thank you, Vincent. Thank you so much for coming. And Bill Yake, I see you're taking some help for your your good body there now. We wish you all the well and get well soon. And thank you for joining us in the rigors of your current existence.
Okay, any other questions? Just a lot of thanks in the chat. So, okay, well, thank you so much,
Hey,
Bob.
don't let the chat go away yet. I want to read the chat before I go, okay?
Okay, well, we can stay on. So if anybody has any questions, fire away. We can just stay on and chat. You can look at the chat function, Bob. I do have one question.
Anybody can go if they want to go. I'm just going to stay for a minute and look at the chat.
Sure, that's fine. For open discussion, so chime in if you have something to say. I do have a question about the change in the genus from Lycaena to Tharsalia.
John, would you speak to that, please? Is John still here? Oh, we might have lost John. Well, I can't speak to it very well, but these are the kinds of changes that we see. And we think John's just messing with us, right? By changing names that we've known for so long. It's irritating, but it's not, what's the word I want? It's not done for nothing. I can't think of a common word. Does that ever happen to you? Come on, help me out here. What's a word that means that you do something that doesn't really mean anything? It's just for no real purpose.
Well, you can call it frivolous, but I think when you expand what you know about species and subspecies, you're gaining information that over time might come to be really important.
That's the point, Kathleen. Yeah, frivolous wasn't quite the word I was looking for, but it'll do. What John does when he changes a name or when he recognizes a different name in the catalog is not frivolous, is anything but frivolous. It is profoundly serious because, as Kathleen said, and that's exactly right, this is about knowledge. This is about learning truths, truths of nature. And now, are those truths somewhat mobile? Are they able to change over time? Of course. Science is like a bed. It's, you unmake it and you make it again. You commit to it, but it doesn't mean you can't change your mind. That's the scientific method. In fact, we proceed in science by trying to not to prove things, but to disprove them, to show that our hypothesis is incorrect. And it better be incorrect, or if it's not, then we adopt it as theory. So with Tharsalia and Lycaena, in the first place, the easiest thing that can happen is that the Code of International Zoological Nomenclature, which John follows to a T in the catalog, says basically that the oldest name that was given to a particular organism is the one that shall be used. So sometimes people just find an older manuscript, an older book. Hey, he named this before she did. So, okay, we'll change the name for that. That's not always done. Sometimes the newer name is kept for stability, but it requires a certain amount of shifting around among the rules and people making that decision. But much more often the changes come about because the lineages are known to change through DNA work these days. So when we learn, either through ecology and just morphology, what they look like, or through genetics, that this animal and that animal are not as closely related as we thought they were, perhaps they do lie in different genera from one another. Lycaena, and actually I think this does relate to what John was just saying. It turns out that, yeah, of course it does. I can answer this question. Lycaena was named for Lycaena fleus, which we call the American copper, which is pretty funny because it was named in Europe, and in Britain it's called the small copper. Ours is called the American copper. It seems to have come across the Beringian land bridge, occur in both places. It doesn't occur in Washington. It occurs just a little bit in the Hiawatha Mountains in Oregon. It occurs in the Sierra Nevada, and it occurs in the Northern Rockies in the Beartooths. And then it occurs on the East Coast as a lowland butterfly, very bizarre. But that butterfly, it turns out, is closely related to Actually, the same species occurs in Eurasia. The other one that does is, did John say the other one was Gubrius? Yeah, our lustrous copper, which is only up on Slate Peak, as far as we know, and a few nearby places in Washington. Then it's down in the Sierra Nevada and the Rocky Mountains. Very, very bright copper, very, very different from the others. Well, it turns out that that has a close relative in Eurasia too. But what John has just informed us is, and I didn't know this, that the DNA studies have now shown that those are the only two species of coppers in Eurasia that bear any close relationship to any New World coppers. Therefore, since they're in the genus Lycaena, which is named for them, all of our coppers have to drop out of Lycaena. They're not in the same lineage, except for the lustrous copper and the American copper. They remain in the genus Lycaena. If you check the catalog, I'm sure that's the case. But all the other coppers are now Tharsalia. They even have subgenera that breaks them down farther. Mariposa is along with the purplish coppers in the subgenus Epidemia, but don't worry about that. So that's why Paulette, it's a matter of increased knowledge, as Kathleen says, through greater tools, in this case, extremely sophisticated genomics, looking at their gene pools and finding out who their common ancestors are. That's what we can actually see in a genomic diagram or a cladogram. We see who their ancestors are. We see where the divergences occurred evolutionarily. Judgment is still called for in some cases. It doesn't answer every question perfectly by any means. It's not a black box that does that, but it will answer questions like that. And that's why the American coppers, except for those two species, one of which is called American copper, had to go back into the old American genus Tharsalia, which was named in America in the 19th century. Does that make sense?
Yeah, that's very helpful. That's exactly what I was looking for. Is just this last answer that you gave about the European species and the American species.
Yeah, and so the knowledge can be irritating when we have to change names. I mean, can you believe that falcons are no longer hawks? They're closer related to parrots, yeah, than they are to hawks. We used to call the whole order of hawks Falconiformes after the falcons. That won't work anymore. Because we know that they are convergently evolved with hawks from different ancestors, that the contemporary raptors are polyphyletic. They have more than one origin. It's been suggested that humans are polyphyletic, but I think we're a long way from that. I don't think so. But when we know that we do have common ancestors, then we can do that. And so Kathleen is so right that we can't do conservation right if we don't know what these things are and where they are. And again, if you look at my map and you see how very, very at risk these coastal populations are, well, we simply wouldn't know that unless we'd been messing around with this name business. So the names are anything but frivolous.
Okay, well, why don't you go ahead and look at the chat? I can't end the meeting until because once I close out the meeting, I think the chat goes away as well. So why don't you look at that? And Kathleen, I was wondering if you could, you just posted a message in the chat about exploring 3 dry lakes around Oahu. Can you tell us a little bit about where, what you're affiliated with and what your plans are, what you're thinking about?
I'm going to bump in before she does and say that Kathleen said in chat that Tia had the sharpest eyes. She was amazing to be in the field with. Tia and Kathleen were field partners on some Spartina studies on Long Island. She sure did, Kathleen. Thanks for mentioning that. Now to you.
Yeah, well, I'm a, sometime butterfly person, pretty inept at it still. I have really crappy eyesight, but I'm a botanist and I really like being in the field. And I really like learning about the animals that use the plants that I've spent my life studying. I live on Willapa Bay, about 16 miles north of Ilwaco. And what intrigues me about the old butterfly sightings in that area is that there are, right in the town of Ilwaco itself, there are 2 remnant lakes they're called dry lakes now, but they're actually still so damp that they're very boggy. There's moss in them and a lot of sedges and stuff. And then east of Owako, there's a third lake that's actually right by the Owako Cemetery. And that's a very interesting area because it also has a relic population of a rhododendron. And our native rhododendron isn't thought to live anywhere within an arc from Grays Harbor down into the Tillamook area. And yet here right at the mouth of the Columbia River, there's this dry lake with native rhodies growing around the edges.
And where is that, Kathleen?
It's right by the Owako Cemetery.
Good gosh.
So I really, really, really want to get into that lake. It's a fascinating spot. It's all privately, there's no public access. It's all privately owned around it. Part of it was logged. We had great access years ago, but that was logged. And now we have huge blackberry thickets to wade through. But I'm working on it on a straightforward way to get us back in so that we can survey this lake. And Bob, what I'll do is I'll pull a Google Earth image and send it to you so you know the areas I'm talking about.
Great, Kathleen. Thank you.
Yeah,
So
thank
does
you
that-
for doing this.
So Kathleen, does that involve like contacting landowners or what, how do you go about it? I'm just curious.
That's exactly how I do it. I contact landowners and I just kind of work through, I use Tax Sifter to find out who the adjacent landowners are.
Mhm.
And I just work through the landowners until I can find somebody who's comfortable with us having access. It just takes a while. Not everybody wants you to be on their property. They're all afraid if they find some rare thing that they're going to be barred from doing anything with their land,
Yes.
except it's already wetland. So that's
Yeah.
already pretty restricted anyway.
Yeah.
It's just a slow process of contacting people and being as unthreatening as possible.
All right.
Just saying, we're curious. We just want to know what's there. And can we come out in July? Because of course, for the butterfly, that's probably, well, late June, early July is probably the best time to be looking.
And always be nice to them.
Oh yeah. Yeah. You have to be nice to them and you have to praise them for having such a beautiful wetland.
Yeah.
Kind of, it makes them feel proud they've got it. And then it makes them wonder why they don't think wetlands are valuable. So I like that. It's got a dual purpose.
I just have to say, Kathleen, that reminds me of another time I got kicked out of a bog. I was near the Rocky Mountain Biological Lab in Colorado with Scott Hoffman Black, the director of the Xerces Society. It was on my butterfly big year. We'd been up to Gothic, the Rocky Mountain Biological Lab, to see Gillette's checker spot, which Paul Ehrlich had introduced there a long time ago. And I was down near Crested Butte, where I used to go as a kid, butterfly collector. And I was in this beautiful little wet meadow, really. It was a wet meadow where there was a big colony of Mormon fritillaries, and they were all roosting communally. They're going to bed in the tall grass, in the tall wet grass communally. I'd never seen that before. And so I was going out there to check it out. And this nearby landowner comes tromping out there. "What are you doing out in my meadow?" And I said, "Sir, it's just a wet meadow. I'm not hurting anything. I'm just looking at these butterflies." And I didn't see that it was posted. And he pointed to a sign. There's a sign right over there. And I was on the other side of the sign.
Right.
So I'm afraid I used Woody Guthrie's line from the last, that verse of "This land is my land" that you never hear. The verse that says, "This land is your land, this land is my land." Let's see. Something about a sign, "But on the other side, it don't say nothing. That side is made for you and me." So I told the landowner, I said, "There's nothing on this side of the sign." That didn't work at all.
Yeah. Yeah. Well, we're glad you're still here.
Yeah, after a couple of those, I am too. No, you always got to be very, very considerate of landowners, for sure. And it's not get your dander up, and and for better or for worse, they're the stewards of what it is you're caring about.
Yeah,
And
absolutely. And if you can make them care about it, that's great.
that's the whole thing right there. Yep.
Yep.
Thank you all. This is a wonderful butterfly. I'm so glad that I bet you all loved it already, but I hope you love it even more now. Next time you see it out there, think about it. Think of these people, these wonderful women in Lepidoptera and others that we have had the honor to name them for, and the Native Americans on whose land they occur, whom we've meant to honor as well. And thank you all for joining me tonight with the Mariposa Copper.
Thank you, Bob.
Thanks very much.
Yep.
All right, see you next month. It'll be a great one next month too.
Nighty night.
Bob, do you want me to leave the chat up a little bit longer?
No, thank you. I've read them now. Thank you very much.
Okay, that sounds great. Okay, well, thank you again, Bob. Thank you so much for giving us such a great talk. I know, well, there were over 60 people at least that were on the meeting tonight, and we
That's
recorded it. And I know that a number of people will be seeing it tomorrow or the next day.
I'm so glad. I'm glad to see Bill Yake's handsome visage there. I'm so glad to see my field companion, David Branch, is here. We go off on butterfly hunts every year. We're going to go back and look for Aphrodite fritillaries again next summer, aren't we, David? We were going to go to Chile in January, but somehow I think that's probably not going to happen. And then Nancy, boy, you've been a great supporter of all this over there in the east side now. Yeah, I'm glad we had east side folks with us as well tonight. So thank you all for coming.
Okay, thank you. We'll see you next month then. I'm going to end the meeting now. All righty. Bye-bye.
Bye-bye.